The specimen jar was too small. At 10,935 meters, the ROV's collection arm grabbed what looked like a translucent sphere the size of a cantaloupe. But it wasn't a colony, it wasn't a cluster, it was a single cell, one organism. Cell biology says this is impossible. At that scale, diffusion fails. The nucleus can't communicate with the outer membrane, surface tension should tear it apart. But there it sat on the bottom of Challenger Deep. And when they brought it to the surface, it didn't collapse. It didn't rupture. It just stopped moving, like it was waiting for 1,100 atmospheres of pressure to return. In biology, there is a rule called the square cube law. It dictates the maximum size a cell can grow before it dies. As a cell gets bigger, its volume increases faster than its surface area. Eventually, the membrane can't absorb enough nutrients to feed the interior, and the cell starves. This is why bacteria are microscopic. This is why human cells are invisible to the naked eye. The math sets a hard limit. A single cell cannot be the size of a football. It physically cannot sustain itself. But in the Challenger Deep, the deepest point on Earth, the rules of biology seem to be suspended. For decades, marine biologists reported seeing ghosts on the sediment, pale, structured shapes that looked like sponges or debris. They ignored them. They were looking for fish. They were looking for animals. They didn't realize that the debris was alive. They were looking at single-celled organisms that had grown to monstrous proportions. Organisms that defy diffusion, defy pressure, and defy classification. Tonight, we are looking at the giant xenophyophores of the Mariana Trench, the single cells that act like architects. To understand why this discovery is so disturbing, we have to go back to a mystery that has haunted oceanography for 62 years. On January 23, 1960, Jacques Piccard and Don Walsh made history. They piloted the bathyscaphe Asterisk Trice to the bottom of the Challenger Deep, 10,911 meters. They spent only 20 minutes on the bottom, but in those 20 minutes Piccard claimed to see something. He looked out the thick plexiglass viewport and radioed the surface. He said he saw a flatfish, a vertebrate, sole or flounder-like, about 30 centimeters long. This observation became legendary. It proved life existed at the bottom, but biologically it was a problem. Fish biochemistry has a hard limit. It's called the TMAO limit. Trimethylamine N-oxide is the chemical fish used to keep their proteins from crushing under pressure. But at depths greater than 8,200 meters, the concentration of TMAO needed to survive would make the fish's cells saltier than seawater. Osmosis would reverse. The fish would die of thirst instantly. No fish has ever been caught below 8,178 meters. The laws of biochemistry say Piccard was hallucinating, or he saw something else. Something flat, pale, and roughly 30 centimeters long, something that looked like an animal but wasn't. For decades, skeptics said Piccard saw a sea cucumber, or a plastic bag, or a trick of the light. It wasn't until unmanned ROVs returned to the site that we found the real culprit, and it was far stranger than a fish. The organism is called Asterisk syringomena fragilissima asterisk, the fragile sandpipe. It is a xenophyophore, a type of foraminifera. These are usually microscopic protists, the kind of thing you look at under a high-power microscope in a high school lab. They are single-celled. But the specimens in the Mariana Trench aren't microscopic, they are macroscopic. Massive, some measure 20 centimeters across, that is 8 inches. Imagine a human blood cell expanded to the size of a stadium. That is the scale of violation we are talking about. How does a single cell get that big without starving? The math says diffusion shouldn't work. Oxygen takes too long to travel from the membrane to the center. The xenophyophore cheats. It isn't a simple balloon of fluid, it is coinacidic. It has multiple nuclei scattered throughout its body, thousands of command centers floating in a shared cytoplasm. It is one cell, but it acts like a city. But the real impossibility isn't its size, it's its behavior. Single cells are supposed to be passive. They drift, they absorb, they divide, they don't build. Yet the xenophyophore builds. It constructs a test, a rigid shell made of sediment particles glued together with its own secretions. And it doesn't just grab random dirt, it selects specific grains. It engineers a structure that is fragile but rigid, a structure that stands up against 16,000 pounds per square inch of pressure. This is engineering, performed by an organism without a brain, without a nervous system, without hands. In 2012, James Cameron returned to Challenger Deep in the deep-sea Challenger Asterisk. His cameras scanned the bottom. He didn't see fish, he saw the ghosts. Field after field of strange, clumped structures on the sediment. He described the bottom as lunar, desolate, but it wasn't desolate, it was inhabited by giants. In 2017, researchers from JAMSTEC, the Japan Agency for Marine Earth Science and Technology, dropped a lander with a baited trap to 10,900 meters. They weren't just looking, they were collecting. When the lander returned to the surface, the traps didn't contain fish. They contained mud, but when Dr. Hidetaka Namaki sifted through that mud, he found them. Intact xenophyophores, 18 centimeters wide. They were delicate, brittle. The test crumbled if you touched it too hard, but inside the sediment shell, the organism was alive. This confirmed Pickard's mistake. The flatfish he saw in 1960 wasn't a sole, it was a xenophyophore. The lights of the asterisk tryst had caught the pale, flat disk of the organism, and his brain, desperate to see life, interpreted it as a fish. But this answer just created a bigger question. If the bottom of the ocean is dominated by single-celled giants, what are they doing? They aren't just sitting there. Recent footage from the 5 Deeps expedition shows high densities of them, up to 4 per square meter. They are farming. The structure the xenophyophore builds isn't just a house, it's a trap and a farm. Dr. Lisa Levin from Scripps Institution of Oceanography has studied how these structures interact with the water. The intricate, folded shape of the shell creates turbulence. It slows down the water current flowing over the sediment. This causes food particles, marine snow, organic detritus, to drop out of the water and settle on the organism. It is passively engineering its environment to bring food to it. But they also hoard. Xenophyophores have been found with extremely high concentrations of heavy metals inside them, lead, uranium, mercury, levels that would be toxic to any other life form. Why, we don't know. Some theories suggest they use the metals to reinforce their structure. Others suggest it's a byproduct of their metabolism. But the most disturbing theory is that they are farming bacteria. Inside the folds of their shell, researchers have found colonies of bacteria. The xenophyophore excretes slime that traps the bacteria, feeds them, and then, when the colony is big enough, absorbs them. It is a single cell that keeps pets, a single cell that runs a ranch. This level of complex behavior, farming, engineering, structural construction, is usually associated with complex animals. Insects, crabs, humans. To see it in a protist is like finding a bacteria that builds a skyscraper. There is one final impossibility: the pressure. 1,100 atmospheres, 16,000 pounds per square inch. At this pressure, water molecules are forced closer together. This changes the way proteins fold. Proteins are huge molecules that need to be a specific shape to work. Pressure distorts that shape. It forces them to unfold, to denature. This is why fish can't survive below 8,200 meters. Their proteins stop working. But the xenophyophore survives at 10,900. Dr. Douglas Bartlett at Scripps calls this pisophilia, pressure-loving. These organisms have evolved proteins that require asterisk crushing pressure to fold correctly. If you brought a xenophyophore to the surface quickly, it wouldn't explode like a deep-sea fish, which dies from expanding gas. It would die of decompression sickness at a molecular level. Its proteins would fall apart because the pressure holding them together is gone. This is why studying them is so hard. You can't just bring them up. You have to bring the pressure up with them. But in 2017, JAMSTEC managed it. They brought a sediment core containing living xenophyophores to the surface in a pressure-retaining chamber. They kept it at 1,100 atmospheres. And they watched. In a laboratory at the University of Tokyo, inside a thick titanium vessel, there is a piece of the Mariana Trench. Inside that vessel, at 1.5 degrees Celsius and 16,000 psi, the xenophyophores are still alive. They don't move much, they don't have eyes to look back at the scientists. They sit, they wait, they filter. They are a reminder that our definition of complex life is biased. We think complexity requires a brain, we think it requires organs. But at the bottom of the world, in the dark and the cold, a single cell has figured out how to grow to the size of a football, build a house out of sand, farm its own food, and survive pressure that would crush a nuclear submarine. It didn't need a brain to do it. It just needed 100 million years of silence. If you want to see the footage of the JAMSTEC lander collecting these giants, click the video on the right. And if you think there's something else hiding in the sediment that we haven't found yet, hit the like button. I'll see you in the dark.